Hi friends, in this video I'm going to show you how to perform a protein or nucleic acid BLAST search at home. Let's get started. So first off, we need to go to the website of NCBI and click on the link in the description below. And here you can see all the different programs that they have developed for bioinformatic analysis. But today we are going to focus on BLAST. So let's click on the link for BLAST. So now we are on the page where we can perform a protein or nucleic acid BLAST search. The one that we're going to do today is the Protein BLAST. We will be clicking on the link for Protein BLAST. Okay, so now we are here. This is the page where we can actually perform the BLAST search. So here on the left, we have some options for the type of search that we want to do. You can either perform a protein query against a nucleotide database, or you can perform a protein query against another protein database. But today, we're going to focus on the most popular one, which is Protein BLAST. Okay, so here you have two options. You can either paste your protein sequence or you can upload a file with your protein sequence. Now, if you want to upload a file, make sure it is in FASTA format, and if you don't know what a FASTA file is, you can watch my previous videos about how to extract protein sequences from different tools like XPC and UniProt and save them in FASTA format. For today's demonstration, I'm going to be using a protein sequence from UniProt. So I already know the ID of the protein, and here you can see I have copied the sequence, and now I'm going to paste it into the box here. Now, before pasting the sequence, you should know that the maximum length of a protein sequence that you can submit for a BLAST search is 10,000 amino acids. If your protein sequence is longer than that, then you might want to divide it into smaller segments and analyze each segment separately. Now you can also select the organism of interest and type the name of the organism here. For today, I'm not going to select anything because I'm going to be performing a BLAST search for a human protein in all organisms. So now I'm going to press enter. Okay, so now you can see that we have entered our protein sequence, and now we have to select some options here. So the first one is the program we want to use. Since we are performing a protein BLAST, we will be selecting the option for Protein BLAST. For the database, you can choose from different databases depending on your needs, but since I'm going to be performing a general BLAST search, I'm just going to select the option for all non-redundant proteins. Now for the word size, this is optional, but generally you can leave it on the default value. Then comes the most important part, which is the expectation value or E value. This is basically the threshold for the significance of matches. The lower the E value, the more significant the match is. Generally, you can set this to 10 to see all the significant matches, or you can reduce this number to see more stringent results. Next is the identity, which is basically the percentage of identical residues between the query sequence and the subject sequence. You can also leave this blank, and it will show you all the percentages. The next one is the gap open and the gap extension penalties. These are used to penalize the introduction of gaps in the alignment. You can leave these on their default values. The next one is the mismatch penalty, which is the penalty for a single mismatch between the query and the subject sequence. You can also leave this on its default value. The next one is the scoring matrix, which is basically a scoring system used to score the alignments. You can choose from different matrices depending on the type of sequences that you are analyzing. Since I'm working with amino acid sequences, I'm going to leave it on BLOSUM 62. Finally, the filter option is used to filter out low-quality hits. You can turn it on if you want to reduce the number of hits, or you can turn it off if you want to see all possible hits. Now that we have selected all the options, we are ready to run the BLAST search. So we're going to press the button for Create a New Search, and now you can see that our BLAST search is being performed. Okay, so now our BLAST search has been completed, and you can see the results here. The first thing that you will notice is the query cover, which is the percentage of the query sequence that has been covered by the alignments. So here, 99% of the sequence has been covered. Then you have the query ID, which is the accession number of your query sequence. Then you have the subject ID, which is the accession number of the subject sequence, or the database sequence that has been aligned to your query sequence. Then you have the percent identity, which is the percentage of identical residues between the query sequence and the subject sequence. Then you have the E value, which is the expectation value, or the significance of the match. Then you have the length of the subject sequence, or the database sequence. The bit score is basically a scoring system used to score the alignments. A higher bit score indicates a more significant match. Finally, the start and end positions indicate the starting and ending positions of the aligned segment in the subject sequence. So let me give you an example. So here you can see the query ID is NP00073, which is the accession number of the human hemoglobin subunit alpha-1 gene. And here you can see the subject ID is XP002884317. This is the accession number of a predicted protein in Danio or zebrafish. And here you can see the percent identity is 92%, which means that 92% of the residues are identical between the query sequence and the subject sequence. Then you have the E value of 0, which indicates that this is a very significant match. Then you have the length of the subject sequence, which is 141 amino acids. The bit score is 195, and the start and end positions indicate the starting and ending positions of the aligned segment in the subject sequence. Now let's say you wanted to download these results in a table format, so you can simply click on this button here to download the results as a table. Okay, so now you can see we have downloaded the results in a table format. The first thing that you will notice is that the columns are actually the same as the ones that you see here. So you have the query cover, query ID, subject ID, percent identity, E value, length, bit score, and the start and end positions. Now let's say you wanted to visualize the alignments, so you can click on the links here to visualize the alignments. So let's click on the first link here. Okay, so now you can see the first alignment. You can see the query sequence here and the subject sequence here. The red color indicates the identical residues, and the green color indicates the conserved residues. So as you can see, this is a very good alignment, and this is a significant match. Now let's say you wanted to see the alignment for the second hit, so you can click on the link here to see the second alignment. Okay, so this is the second alignment. Again, the red color indicates the identical residues, and the green color indicates the conserved residues. So this is also a good alignment, and this is also a significant match. This is all for today's tutorial. I hope you found this video useful. If you did, please hit the like button and subscribe to the channel. Thank you for watching, and I will see you in the next video.